In late 2023, history wasn't just made, it was edited. The first ever approval of a CRISPR-based gene therapy, called Casgevy, marked a quiet turning point for medicine. This wasn't about a new pill or a surgical procedure, it was the authorization to rewrite a person's own biological source code to cure a disease. The targets were sickle cell disease and beta thalassemia, lifelong conditions stemming from a single error in the gene that builds hemoglobin. But the therapy doesn't actually fix that error. Instead, it performs a clever piece of biological trickery by awakening a ghost within our own DNA, a silent, superior gene we all possess but forget how to use shortly after birth. Hemoglobin is the protein that makes your blood red and, more importantly, carries oxygen from your lungs to every other cell. In sickle cell disease, a single typo in the hemoglobin gene creates a faulty version that clumps together, deforming the red blood cells into rigid crescents. These misshapen cells get stuck, causing immense pain and organ damage. But here's a strange fact. As a fetus, you didn't use this version. You used fetal hemoglobin, or HbF. It's more efficient at capturing oxygen, a useful trait in the womb, and, crucially, the sickle cell mutation has no effect on it. The gene for HbF is perfect, even in people with sickle cell. After birth, a genetic switch flips, turning off the fetal version and turning on the adult one. What if you could just find that switch and flip it back? That switch has a name: BCL11A. It's a gene whose entire purpose is to act as a repressor. Think of it as a molecular security guard. After you're born, the BCL11A protein is produced, travels to the fetal hemoglobin gene, and physically blocks it from being read. It silences it. The clue came from nature itself. Scientists observed that some individuals with sickle cell disease had almost no symptoms. When they looked at their DNA, they found these people had a naturally occurring, harmless mutation that made their BCL11A gene less effective. Their security guard was asleep on the job, allowing their bodies to continue producing high levels of protective fetal hemoglobin throughout their lives. This is where CRISPR-Cas9 enters the story. The process begins by taking a patient's own blood stem cells out of their body. In a highly controlled lab, the CRISPR machinery is introduced to these cells. Its job is not to repair the original sickle cell flaw, but to find the BCL1A gene. It is guided to a very specific spot, not the part that makes the protein, but an enhancer region that acts as the volume knob, telling the gene how loudly to express itself in red blood cells. There, the Cas9 enzyme makes a single, precise cut in the DNA, breaking the enhancer. The cell's own DNA repair system rushes to fix this cut, but the repair is often imperfect, leaving a small mutation or scar that permanently disables the enhancer. The security guard's instructions are now garbled. After this editing process is complete, the patient undergoes chemotherapy to clear out their old, unedited bone marrow. Then, their own modified stem cells are infused back into their bloodstream. As these edited cells take root and begin producing new blood, something remarkable happens. With the BCL11A repressor effectively neutralized, the fetal hemoglobin gene awakens from its slumber and roars back to life. The new red blood cells are now hybrids. They still produce the faulty adult hemoglobin, but they are also flooded with the powerful, functional fetal hemoglobin. This high concentration of HbF physically gets in the way, preventing the faulty adult proteins from linking together and causing the cell to sickle. The disease's primary mechanism is defeated. This is more than a cure for one disease. It's a demonstration of a profound principle. We carry within us an entire library of dormant genes from our evolutionary and developmental past. The most elegant solution might not always be to fix what's broken, but to reawaken something powerful and perfect that we've simply forgotten how to use. Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.